Hello, everyone who is already here. My name is Albert Laszlo Barabashi, and we're doing something that we have never had a chance to do before, but adapting to the 21st century, we're doing effectively a German part to how do we actually identify drugs that could be effective for patients infected by the COVID-19 virus. Now, the traditional approach for finding drugs is to develop new ones that are specifically tailored to the disease. Well, what we know is that the traditional approach of iterative development, experimental testing, clinical validation, and approval of new drugs is not at all feasible in the current environment, in a sense that the timescales necessary to put a new drug on the market is really measured in years and not in weeks or months that we really need a response for. So a much more realistic strategy relies on drug repurposing, which effectively means that we try to find drugs that are already on the market, already been given to patients for different conditions that may also have a therapeutic effect on the COVID-19 patients. So, and the reason we do so is because these drugs already have well-defined toxicity and side effects. So if they happen to be effective against COVID-19, then they can be really fastly deployed and used in patients. Now, the reason we're actually focused on this problem is because in the last decade, network medicine as a discipline, and I'll talk a little bit about, has developed and validated a series of computational tools that helped us to identify drug repurposing opportunities. And really what we're going to be doing in the next 30 to 40 minutes is to tell you how we adapted these tools to the COVID-19 disease and effectively to talk about some of the results we obtained through this process. Now, let's talk for a second about network medicine, which is network medicine is really an intersection of network science and medicine, and has really developed in the last decade with the purpose of acknowledging that none of the genes and none of the proteins in our cells really act alone, but they really work in tandem with others by interacting with many other proteins and metabolites and so on. And that if you want to understand how the cell works, you need to really map out and understand the role of these interactions. And second, equally important, if we want to understand disease, which is typically implies the breakdown of this network, we need to think in terms of the network and not only in terms of a disease gene or mutation, but in terms of the impact that mutation has on the integrity of the network and its ability to carry on its basic functions. So network medicine is now a fastly maturing discipline. For example, Harvard University has now a network medicine division that was started by Joe Loscazo and many others on this call. And really, we're deploying a series of network-based tools to try to understand human disease. Now, network medicine provides us a series of tools that can be immediately applied in this context. One of them is to identify the disease module. The disease module really relies on the observation that the disease genes are not randomly distributed in the network within our cells, but rather they tend to agglomerate in a certain network neighborhood. And that's what we call disease module, and we'll talk about that in this context. And once you know the disease module, you are in a better position to identify disease genes and to kind of understand the disease mechanism. But once you have the disease module, you can also talk about drug target identification and repurposing opportunities, which practically means that you are looking for drugs that happen to be hitting in the neighborhood or within the disease module. And we'll explain how that works. Once you could also use these tools to identify drug combinations, drugs that work synergetically together to kind of help alleviate the symptoms, and offer us tools to kind of think in personalized manner to identify for a particular patient which part of the disease module is being disrupted and what are its consequences. And one day may offer us to even control some of these subcellular networks, which may lead to the cure or some other benefits for the patient. Now, network medicine has typically focused on complex diseases, but my lab and many other labs were part of a major collaboration in the last decade, a big NIH center that focused on developing tools for exploring virus-host interactions. And this was done with the leadership of Mark Vidal at Dana-Farber Cancer Institute. And it was a project that focused at that time on cancer-causing viruses, EBV and HPV. And it had a major experimental as well as a modeling theoretical component. The experimental part was the goal to experimentally identify which EBV protein, which is the viral proteins, interact with what human proteins. That is, in the language we're going to use, we have to identify the viral targets. These are the proteins to which the viral proteins bind and down the line affect the human protein's ability to carry on its basic function. And by also mapping these human proteins through interactions to other human proteins as well as to disease proteins, we were able to identify the network neighborhood where the viruses are hitting. And that gave us information about the molecular processes that are disrupted by the virus and helped us identify the disease symptoms. So this was a methodology that we developed for cancer-causing viruses, but obviously this is very helpful now when it comes to the COVID infection as well. Another development is very useful in this context, which is a series of tools that have been developed on drug repurposing. And the idea of drug repurposing is relatively simple. Drugs who may not be subscribed for the given condition, if they have targets in the vicinity of the appropriate disease genes, may be disrupting the network and the disease genes in a way that is beneficial to the patient. So the methodology of drug repurposing effectively is based on the idea that we map the targets of all of the existing drugs on the network of the human interaction, the protein-protein interaction between the human cell, the proteins. We also map the disease genes, and we look for drugs that hit in the proximity of disease genes. And we'll talk a little bit about what proximity means in this case. But the expectation is that drugs with targets in a network vicinity of a disease module helps or are potential drug repurposing candidates. And there are network medicine-based tools, but there are also AI tools that allow us to do that, and we'll talk about that. Now, let's go back to COVID-19, or let's start talking about COVID-19 and see how we can actually pull these tools together to talk about drug repurposing candidates in this context. So the language we're going to use is the following. So we have actually the virus, which is shown here on the left. And the virus is generating about 29 different proteins that are in the human cell. These are viral proteins, but once they are generated in the human cell, they bind to existing human proteins. So the first interaction we care about is the viral protein, human protein interaction. And these will define what we call the viral targets. The viral targets are the human proteins that the virus proteins bind to. We will also be dealing with the human protein interaction network. That will be most of the interactions in the large network that we'll be talking about. That is simply what kind of interactions are experimentally known between the human proteins. What we will see is that the viral proteins hit in a certain network neighborhood of this very large network. And that neighborhood we will call the COVID-19 disease module. And the last piece of the puzzle are the drugs. The drugs interact with the human interactome or the subcellular network by targeting some of the human proteins. These are shown in dark green over here. And what we know from previous studies as well here is that they define what we call the drug module, the drug disease module. That is, what is the neighborhood that the drug is really targeting? And what we will see is that the drugs are efficacious against this particular disease. So they work for that disease if their drug module overlaps with the disease module. That is, in this case, the drug targets hit in the immediate vicinity of the COVID-19 disease module. So that will be the program that we will try to execute, and we'll be searching for drugs that have this particular property. How are we going to do that? The input data is the human interactome. This is all the experimentally known interactions between the human proteins. We're talking about 18,000 proteins and 300,000 pairwise protein-protein interactions between them. The other input data for our study is the SARS-CoV targets that really comes from the work of Gordon et al. Or the Krogan lab, who on March 22nd put out the first experimental assay of identifying what are the human proteins that are targeted by the virus. And finally, the last input data will be the drug targets, which is we're looking at 7,500 drugs from Drug Bank that altogether have about 4,100 targets. Now, there are fewer targets than drugs, partly because many drugs target the same target. And there are also drugs that do not have a known target. Those that don't have a known target cannot be studied with our methods. Now, let's talk about the methods. We will deploy 3 different methodologies for drug repurposing, one based on network proximity, one on network diffusion, and one on AI. Each methodology provides different ranking lists. So these are what we call pipelines or predictive pipelines, and you see how many we have from each methodology. And the outcome of this will be, on one end, actually, what are the potential drug repurposing candidates? And we'll talk about how we combine these different predictions. But these methodologies also give us information about what are the tissues that could be potentially infected by the virus, and as well potential comorbidities of the infection. Now, let's get started on that. And the first issue we have to talk about is the disease module concept. What you see here is actually an artistic representation, or actually a real mapping of the human interactome. And in color, I'm actually showing you all the proteins that are involved in a disease, in this case, asthma. And what you see that in this large network, the asthma proteins are not randomly distributed, but they tend to agglomerate in a certain network neighborhood. That network neighborhood defines the disease module. And the first step we need to do is to identify this disease module for the COVID-19 infection. To do so, we took the 332 proteins that the Krogan lab has identified that are being targeted by the virus, and we mapped them into this network of 18,000 proteins. And what we found is that 280 of them, actually I went too far, what we found is 208 of them for one giant component, or we call the LLC, the large connected component, which is shown here on the left, and the rest are actually forming small subclusters. These small subclusters may want to be connected to the big one, but we may not have all the interactions, for all protein-protein interactions to do so. The data is never complete in this space. But the first question we ask is that, could this cluster that we see on the left have emerged by chance, or it's telling us something about the network neighborhood where the virus hits? And for that, we take randomly 332 proteins and distribute it on the network with certain controls, what we call degree preserving controls. And we ask, what would be the size of the cluster you would get if you randomly choose these proteins? And what this plot shows is that the randomly chosen clusters would have approximately a size of 180 to 190, and gives us a statistical significance with a z-score of 1.65. What it indicates that this network neighborhood didn't emerge by chance, but it is really telling us that the virus attacks the human interactome in a specific network neighborhood. This is important because it will be this network neighborhood that we're going to see over and over in the presentation, as the repurposing candidates must target proteins in the network vicinity of this network disease module. Now, I'm going to actually pass it on the slides to Daisy, who's a postdoc at Northeastern, who will talk about tissue specificity and in what tissues could the disease express itself. Daisy.
Hi. So we wanted to understand whether this module would be preserved in other tissues or not, and mainly in the lungs, because that's where we have the primary symptoms of the disease, or at least the ones that are mostly associated with the disease. So we mapped the proteins based on whether they were expressed or not in the lungs. And what we found is that actually only 58% of those proteins, they are actively expressed in the lungs. And when we reconstruct the module is much more significant than the overall proteins that we had in the previous slide. So it gives us a z-score of 1.78, and the distribution is around 160. So the average LCC that we expect is around 160. And actually the one that we found is 182. Can you go next, Laszlo?
Yep.
Yep. Oops.
Too far. I'm back.
Thank you. So we saw that we have a module in the lungs, and we want to understand whether other tissues could be also affected by the virus. So we use GTEx as a baseline to know which which genes they were expressed in which one of those 56 tissues. And we found that apart from the lungs, we had multiple brain regions also affected that could be affected by the virus. And it's also in line with some clinical observations, such as loss of smell and taste, headaches, dizziness, seizures. And one thing that is quite interesting here is that in autopsies, they found involvement of spinal cord. And we also found as one of the tissues or the brain regions that is also affected. Similarly, we have the digestive system that many patients, they report having diarrhea, nausea, vomiting, and abdominal pain. And we also found the digestive system as one of the most involved. Similar, we have cardiovascular tissues, and often patients with underlying cardiovascular diseases, they also have increased risk of death when committed by COVID-19. And another interesting finding is the reproductive system. And it's also being reported in some autopsies that prostate and testes are highly involved with COVID. We still don't know the extension of that, but it's quite an interesting finding.
Okay, thank you, Daisy. And so now we actually want to kind of ask that now that we know what tissues could the virus be expressed in, we want to kind of go back and say what kind of comorbidities do we expect? And this is already giving you some information about comorbidities. Let me just give you some background information. One of the things that we learned by studying actually multiple diseases in the framework of the network is that each disease has its disease module. And when two disease modules overlap, there is typically a comorbidity between them. And here we're showing about like 50 to 100 diseases placed in this network space. And if you kind of look very closely, you will realize that that diseases that tend to have similar symptoms and mechanisms tend to be closer in the network space, and those that are unrelated are further away. So therefore, we ask explicitly what diseases could actually show comorbidity. For that, we have a comorbidity measure that measures the distance between the COVID-19 disease module and 299 other potential diseases. And what we find actually is that we recover several of the known comorbidities that has been observed by clinicians. In particular, we see heart disease. So just to interpret this plot, each node corresponds to one of the 299 diseases that we looked at. Here we show the disease classes. The bigger the node, the more genes are associated with that disease. And the closer a circle to the center, the closer it is to the COVID-19. So as you go further away, the further you are from the disease. So the close diseases are the ones that are expected to show comorbidity, and we see their heart disease, but we also see multiple neurological diseases in line with the earlier observation that the virus could express itself in the brain as well. So now it's time to actually go to the real part we are here, which is to say, how do we identify repurposable drugs? And one of the things we learned from this previous slide is that the COVID-19 module does not explicitly overlap with any of the known disease modules. The implication of that is important because it means that we cannot take a therapies already existing for the disease and cure COVID-19 patients. Rather, repurposing strategies should focus on drugs previously approved for other pathogens. So what, I'm sorry, let me start over. So what typically people do is that they take drugs approved for other pathogens, like malaria, or they take drugs that target one of the targets of the virus itself. But one of the things we learned actually during the last decade is that most approved drugs on the market that are efficacious against a particular disease do not target directly the disease proteins themselves, but they bind in their network vicinity, which indicates that just by looking at the direct targets of the virus will actually not allow us to identify all the potential disease, drug candidates. So we are now developing a strategy to identify drugs that may not hit directly the viral proteins or its immediate neighborhood, but they could still have efficacy. And as I said earlier, we're going to use 3 different pipelines, and each of the pipeline is run by a different researcher, and they will all present their results in the next slides. So I'm going to pass it on to Xiao, who is a postdoc again in our lab, and he developed, or he applied to the proximity-based ranking to this particular virus. Xiao.
Hi, everyone. Let me introduce the network proximity-based ranking method. As Laszlo mentioned, previous work in our lab showed that in the protein-protein interactome, drugs with targets proximal to disease genes tend to be efficacious. Therefore, we can rank or predict drugs by using a relative proximity z-score. How this works is that we first compute the average shortest distance, d, of all drug targets to their close disease genes, as you can see from the shortest passes highlighted in red in the figure on the left. And after we got this d value, we convert it into a significance z-score. For this, we randomly select the same number of drug targets and disease genes and compute d for each randomization. We will end up with a reference d distribution, as you can see in the bottom. The relative proximity z-score is then computed from this, the mean and standard deviation of this distribution. In short, this z-score represents how proximal the drug is compared with random expectation. Then drugs are ranked based on this relative proximity z-score. The more negative the z-score is, it means the more proximal the drug is to disease genes, and thus the drug is predicted to be more efficacious. Could you slide, please, Laszlo? Thanks. So to actually compute proximity for known drugs, we obtain drug data from drug bank and then use 3 pipelines here with different targets. The first pipeline uses all targets. The second pipeline disregards targets that are less related to pharmacological effects. The third pipeline computes these scores using differentially expressed genes instead of reported targets. And at last, let us look at a quick example on the right where you see the interactome neighborhood where 2 drugs, chloroquine, etanercept, hits. When we compute the proximity, if you look at the bottom, we got a z-score for chloroquine to be minus 1.82, meaning that its distance to COVID-19 genes is significantly shorter than random, thus suggesting this could be an effective drug. On the other hand, etanercept has a positive z-score, 1.29, meaning that this drug is distant from disease genes and thus is predicted to be less effective. To summarize, we use a network-based proximity measure as a predictor of drug effectiveness. And this concludes the introduction of the network proximity-based method.
Thank you, Xiao. The next one is the diffusion-based models, and Asher Ameli, who implemented that, will tell you about that.
All right, thank you, Laszlo, and hello, everyone. My colleague presented the proximity that relies on the shortest path, and it performs pretty well. Now, what if we take into account the full description of the network with the aim to capture a finer grained distinction of the nodes? We represent each node of the interactome by a vector here, as you see, as HE, that shows the global state of that node in the network. So to represent node I, we calculate how a perturbation initiated from that node impacts other nodes of the interactome. And we can think of multiple options to quantify the similarity between two nodes. One is to calculate the distance-based metrics such as L1 or L2 norm. You simply subtract two vectors element-wise and calculate the L1 norm between them. In this case, if two nodes have identical neighbors, the similarity between them would be 0, and you have reached the maximum similarity. This method is known as DST, or diffusion state distance. Well, one advantage, or I would say disadvantage, of DST is that it is symmetric. However, we know that PPI has an asymmetric nature. And in addition to this, there are also some articles that highlight the drawbacks of distance-based measurements, especially if they have higher orders such as L2 norm. Now, motivated by these 2 points that I just mentioned, we not only calculate the pairwise similarity by the L1 norm of the corresponding vectors, but also by using the relative entropy between them, known as KL divergence, which is asymmetric, and also Jensen-Shannon, which is the symmetrized version of KL divergence. Next slide, please. All right. All I said was to quantify the pairwise similarity of two nodes. Now, the question is that how can we apply this at the drug level to measure the impact of a drug? Because as you see in this cartoon, each drug might have multiple targets at the protein level. And here is our solution. Here is how we measure it. The first pipeline uses the average over minimum DST of drug targets and COVID-19 proteins. The next two pipelines, use KL divergence instead of DST. Pipeline 2 calculates the average over the minimum of KL divergences, and pipeline 3 calculates the average over the median of KL divergences. As you see, we are testing both median and minimum, as the median is robust to the existence of outliers, and minimum may capture the maximum possible signal. Similar to pipeline 2 and 3, we also deploy Jensen-Shannon. So in total, we have 5 pipelines. These metrics are all applied on more than 6,000 drugs of drug bank. Thank you.
Thank you, Asher. And then finally, the third pipeline will be presented by Marinka Zichnik from Harvard University, who developed them. Marinka.
Thank you, Laszlo. Hello, everyone. The third pipeline used AI algorithms to identify repurposing opportunities for COVID-19. So as a first step to that, we used the power of knowledge graphs or heterogeneous networks to really bring together different types of biomedical interactions. So what I'm showing here on this slide is a heterogeneous graph that consists of protein nodes shown at the bottom, then disease nodes in the middle layer, and drug nodes shown at the top. And this unified representation allowed us to integrate in different kinds of interactions, including protein-protein interactions between protein nodes, protein disease, protein drugs, and associations. Given this representation, then the machine learning task becomes to predict what diseases a new molecule or a drug might treat, which corresponds to predicting and discovering new links or edges between nodes in that knowledge graph that represent drugs and diseases. In particular, our primary interest will be then to predict links between drug nodes and the node that would represent COVID-19. So what is actually the methodology then that allows us to predict these drug disease links that would be indicative of treatments? So this is shown in this next slide. And the methodology that we use that allow us to learn and reason over that knowledge graph is known as graph neural network or graph representation learning algorithms. And particularly the method that we deployed here has two main components. So in the first component, we took the knowledge graph from the previous slide and we learned an embedding for every node in that graph. So embedding is the key technical concept here. And what it means is that every node in the graph was represented by a short d-dimensional, low d-dimensional vector, which is known as embedding, where the idea of learning those embeddings was such that nodes in the knowledge graph that have similar local network neighborhood are embedded close together in that low dimensional embedding space. So intuitively, what we did was we represented the combinatorial nature of the knowledge graph with this compact vector embedding space such that performing algebraic operations then in the embedding space reflects topology of the graph. In the second step, we leverage the learned embedding space. We take the learned embeddings for drugs and diseases, and we use them and learn what is the effective way of combining those embeddings in order to predict what diseases a given drug might treat. We train this model on all drug and disease data and their indications, and then use it to make predictions for COVID-19. Next slide, please. So in particular, to generate predictions for COVID-19, we then used 4 different ways that were constructed as 4 pipelines that use different types of decoders, different ways of how we can combine those drug disease embeddings into predictions. Intuitively, what is common to all those pipelines is that we explore the learned embedding space as shown in the right part of the slide, when each point represents a drug or a disease, and they are color-coded. And we see that somewhere in the center, there is the COVID-19 embedding of the COVID-19 disease, and some drugs are very close to that disease in the embedding space, indicating that those drugs have similar local network neighborhoods as COVID-19 disease module. So then in different pipelines, we use different ways of how we can measure and characterize that notion of similarity between COVID-19 and candidate drugs in the learned embedding space. So in pipeline 1, we calculated the cosine distance between COVID-19 and drugs in the decoded embedding space. And then in pipeline 2, 3, and 4, we developed decoders that allow us to either focus on very fine-grained topological structure of the embedding space, or in pipeline 3, or in pipeline 4, really focusing on preserving the broad topological structure of the embedding space. And that has then produced 4 ranking lists that were then integrated to obtain the final predictions.
Perfect. Thank you, Marinka. And now, of course, the big challenge is that how different are these predictions, because they all based on a different methodology. So the first thing we actually ask is that how similar and how different are the 12 different ranking lists that we obtain through the 3 different methods. And we don't expect them to be truly independent, because they start from the same drugs, the same drug targets, they work on the same interactome, and they explore the role of the same perturbations from the virus. So to explore how different they are or how similar, we measure the Kendall-Tau correlation of the rankings between them. And what we find actually is very interesting for us. What is not surprising at all is that, for example, the diffusion methods are very, very kind of similar. The outcomes are similar by the 4 diffusion methods. So are about the 5 diffusion methods, and so are the 4 AI-based methods that are very highly correlated among each other. On the proximity methods, we see a break into 2 proximity methods that are based only on the drug targets that are correlated, and the one actually that kind of uses gene expression data is uncorrelated from the previous one. What the main message here, which is interesting for us, is that while there are correlations within the methods, there are no strong correlations between the methods. And this is important because it indicates that different methods offer complementary ranking information. So the challenge for us, how do we extract these different informations for the benefit of arriving to one ranked list? So for that, we actually used an algorithm that combines the different, well, before that, we want to see how predictive each pipeline is. So we measured the ROC for each pipeline individually against the appropriate ground truth. Now, it's hard to find the real ground truth in this case, but there is one, which is what the medical community thinks that could be the right drug repurposing candidates. And for that, we use 67 drugs that as of last week, according to the clinicaltrials.gov, were in clinical trials for COVID-19 patients. So that's what we use as ground truth. And we ask how the different rank lists predict these drugs. And we're by measuring the ROC curves and the AUC. And what we find is that the best individual performance is provided by the 4 AI-based pipeline that provide of AUC in the vicinity of 0.86, 87. Next in list, so the AI does very well. I was very pleasantly surprised to see that. The second best AUCs are provided by the proximity measures, in particular the gene expression-based proximity. And then the diffusion methods offer somewhat lower AUCs as a group. This was somewhat surprising to us professionally because diffusion methods are global methods, just like the AI. So we expected that the diffusion should be higher than the simple proximity, but the backing order is different. Now, the real challenge, how do we combine these different predictions together? Because we don't want to just pick the best list because there's complementary information in each of the lists. And to do that, we actually use the rank agglomeration algorithm that Marinka and Onur have implemented, shown here, and work together with Daisy to test it. And what this algorithm does is that it looks at all the lists and then tries to maximize the number of pairwise agreement between the final ranking and each input ranking. This is a standard methodology. There are multiple versions of that in the computer science literature. And the first asks, what happens if we combine the measurements from each pipeline? And what we find is that the combined performance of the AI methods is 0.87, which is the similar or exactly the same as the best performance of one of the AI pipelines. But the combined diffusion pipelines actually offer lower proximity as if information was lost. And in the proximity pipelines, however, combining them offers an improvement, partly because there are two different inputs information coming in, one from gene expression and one from just pure proximity to targets. But the good news is this, and that's really essential in this slide. When we combine all 12 pipelines together, we obtain an AUC that is higher than any individual AUC. So this means that this ranking managed to extract new information, and this final ranking with an AUC of 0.89 will really define our final drug repurposing list. At this moment, so let's look at that list, and this is why we waited for so long. We had lots of technical results to explain in the meantime, and now it's my pleasure to pass it on to Joe Loscalzo from Harvard Medical School, who will tell us how he actually took the predictions from these 12 from the final ranking list and pared down to the 86 drugs that we have in the final list. Joe.
Well, thank you, Laszlo, and good morning, everyone. Paring down the list was a bit of a challenge. Importantly, the list was constructed in a totally unbiased way. My job, knowing something about the pharmacology and the drug classes, was to help reduce that list to a more manageable number that might have a greater likelihood of success when tested with respect to its efficacy in limiting cytotoxicity by the virus. So among the different approaches I used to reduce the list, and I focused largely on the top 10% of drugs in the overall rank list, was to eliminate members of the same class that might occur within that pool. I also eliminated those drugs that might have direct effects that could be toxic from my knowledge of the drug's pharmacology in different organ systems, including those that were affected by the virus itself. Remember that this analysis doesn't really tell us anything about whether or not the effect of a drug could be positive or adverse. There's no directionality to its action, at least with respect to this specific disease about which we know so little at this point in time. So my eliminating drugs from the list is based on toxicities that we know to be the case from their use in other settings. The last point is that in some cases I did include drugs that were members of a common class because they're being tested broadly, and we wanted to see where they in fact fell throughout the overall rank list. And if you look at the last drug on this list, azithromycin, you can see that it ranks 786, whereas troliandomycin, which is another member of the macrolide class of antibiotics, ranks third in the list. I included azithromycin because it's in the midst of clinical trials at this point in time. A couple of other points to mention. One is that you may have recognized in a few of the slides that Laszlo and the other speakers showed that many drugs have multiple targets that are known about, and they are often represented in some of the modules that have been depicted in this presentation. In fact, Chartier and colleagues about 3 years ago analyzed from a bioinformatic perspective, the extent to which a binding motif for a given drug is recapitulated throughout the human proteome. And as it turns out, the distribution is very long-tailed with a median of about 32 targets per drug. So it's more the exception that a drug has truly a single target rather than a rule. And in fact, that works to our benefit here because there may also be multiple targets that are not necessarily related to the pathobiology of the disease for which the drug was developed that appears in the available data sets that can then be used to impute their potential role as targets for the drug in the COVID-19 infection. And that's really a reflection of another point Laszlo raised, that the analysis failed to show any significant overlap between known disease modules and the COVIDome, if you will. Despite that fact, there clearly are drug targets used for those known diseases that sit more proximate to the COVIDome than perhaps to the other disease modules that have worked to our benefit here. I'll make one last point because there was a question raised about this point by one of our participants, that hydroxychloroquine was not included in this list, but chloroquine was in the list of 86 that we're now analyzing in cytotoxicity assays. And the reason is that it's a similar drug, although it has a different set of toxicities, and it was ranked lower on the list than chloroquine. So from the standpoint of an in vitro assay, there was really no reason to test both. If we were to get to in vivo assays, and they are ongoing for both of these drugs in humans anyway, we would compare the two because of the differential in vivo toxicities of the two drugs of the same class. Thank you, Laszlo.
Thank you, Joe. And so finally, the question is, how do we know if we are right? And of course, we'll talk a little bit of what tests are coming, but we can do what we call validation studies that rely on existing data. And I'm going to pass on for the next two slides to Italo De Valle, who will tell us some of the validation studies that we already done. Italo.
Thanks, Laszlo. Good morning, everyone. So here we asked whether these drugs could modulate the processes targeted by the COVID-19 virus. And for that, we retrieved experimental data from the connectivity map database of 59 of the 81 drugs that we selected. And this data is organized as expression profiles. So basically, for each drug, we have how the drug perturbs the expression of the genes in cell lines. And we asked the question whether the drug could perturb the COVID-19 targets. So in this figure, in the slide, you can see the COVID-19 module. And the colored nodes are the nodes are the proteins, the genes that the drug mitoxantron significantly perturb. And we can see that 22% of the COVID-19 targets are perturbed by this specific drug. So we applied the same reasoning across the 59 drugs, and we found that 43 of them can effectively perturb the COVID module. And this 43 is much higher than we would expect if we were to select a random list of 59 drugs. So the drugs that perturb the most number of COVID targets in this data set were carfilzomib, flutamide, and bortezomib. And if we limit the expression data only for cell lines from lung, the drugs with the highest overlap would be mitoxantron and ponatinib. So in the next slide, we now make a different question, is whether these drugs could counteract the effect of the virus. So for example, if the drug could downregulate the genes that are upregulated by the virus or vice versa. And for that, we also integrated gene expression data from cell lines from the A549 cell line that was infected with the SARS-CoV virus. And we had the perturbation for 120 differentially expressed genes. So we can, in the plot, in the slide, we can see that for those 120 genes that are perturbed by the virus, the drug bortezomib counteract the effects of 65 of those genes. So we can see genes that are downregulated by the drug and upregulated by the virus and vice versa. And we can summarize this trend with a negative correlation between the two perturbations profiles, the perturbation caused by the drug and the perturbation caused by the virus. And we see a negative correlation for 22 of the drugs that we assessed from the connectivity map database. You are muted, Laszlo.
Yes, thank you, Italo. And finally, now that we presented the results, let's talk a little bit about some disclaimers, right? As Joe has actually very nicely mentioned that some of the data sets that we're using have certain limitations. So we actually ranked the existing drugs based on the expected efficacy on COVID-19 patients, but there doesn't mean that there are no other drugs who could be actually efficient in this space. And our ranking could improve as new data becomes available, like new virus host binding interactions. There are several groups in Europe and the US who are now measuring again this type of interactions. If we would get more extensive microarray data for COVID-19 patients, that would be also a game changer for trying to figure out which drugs are more appropriate. And of course, new drugs could appear in drug bank. They did already actually in response to the COVID and so on. But what I want to emphasize is the methodology is very general and can be applied to any drug or any classes of drugs, whether they're already on the market or they are in the experimental phase. And just to kind of illustrate that, let me show you some examples of what type of results comes from this analysis. This is our ritonavir, our #1 ranked drug, which is already in 20 clinical trials. It was almost none when we actually came up with it originally in the ranking. And the lower this number is here at the AI, it means that the more likely that it predicts. So the AI and the proximity measures are a very big fan of this drug. The diffusion is not so much. But what I want to say is that now we actually can pinpoint the mechanism through which the drug interacts with the COVID-19 targets. So here is the module of the COVID-19, and in red we're showing the drug targets. And here's the blow off of that region. And this particular network maps allows us to make experimentally testable hypotheses of what proteins should be affected by the drug in an infected cell line. So this is not only simply telling us, okay, this drug should be effective, but allows us to say how it should be effective. And if we make an experimental assay, what other proteins we need to test their expression pattern to see whether the drug would or would not be effective in the way we predict. I'm showing a chloroquine because so much interest has been shown towards that. There are 76 clinical trials right now. Actually, almost half of the clinical trials are focusing on this very drug, which actually says, why do we need other candidates? Because everybody's jumping on this bandwagon. And you see that the AI and the proximity are very excited about this drug. And here is the local network neighborhood that is telling us how the drug targets should interact with what kind of COVID targets. And you see particularly here TNF target and a couple of others, right, that are hitting in the direct vicinity of the COVID targets. The third example is lopinavir, which is currently in 17 clinical trials, and it's ranked 146 on our list. And again, we actually get mixed results on that. The proximity is not so sure about that. AI likes it, and diffusion is again mixed. And here's the subgraph. And I wanted to use something that is actually not being predicted to be effective. So this is afitanib, which is ranked 586 in our methodology. Neither AI nor proximity are excited about. Diffusion actually looks favorably at it. And what is interesting is that just looking at the network is hard to really piece out why not, because this drug also hits in the neighborhood of several COVID targets. But probably one of the reasons that the methodologies are not excited about this is because this turns out to be a hub. And when the drug hits a hub, it has actually much less specific consequences on the interaction partners because it interacts with so many other partners. So I should also say finally that what Joe already emphasized, that this methodology doesn't tell you alone, the network-based one, whether the drug will be beneficial to the COVID-19 patients or they actually aggravate the symptoms of the phenotype. And to answer that, we need microarray data, we need cell line-based experiment, human cell line-based experiments that distinguish that. And also, these drugs all have known toxicities, and some of them are relatively limited, so they could actually be moved directly to clinical trial when the case for them strengthens, and we hope people will do that. So finally, let me just say what is next. We are, as we speak, actually waiting for the drugs to arrive, which will then be screened by the National Emerging Infectious Disease Laboratory here at Boston University. But while we're waiting for the experiment to actually start and finalize, we expect readout in about a week after they start, we are also working to understand why each pipeline predicts for each drug what they do. And we really want to get into the heart of the AI and the other methodologies to understand where the signal comes from, how different they are, and where is the common point where they agree on. And I think that's a very interesting journey for us professionally. And when enough new data emerges, we will probably rerun the pipelines with this understanding that we gain in the meantime and see how an improved pipeline compares to what has emerged in the meantime as success or failures in the clinical trials. If you're interested, of course, the manuscript's available on the link, and feel free to contact us if you have any specific questions, you would like to follow up on some of these and you need additional information. I should also say that our work would have not been possible if the Krogan Lab doesn't put out on March 22nd, actually, the human host virus interactions. And we're very thankful for them. And we've been working nonstop since they put that data available. And there are many other efforts that are actually focusing on drug repurposing, like the very visual methodology of Jan Baumbach, who's using at TU Munich. And look at his website because you can actually see the drugs in action. Fei-Shong Cheng, a former lab member, actually has been the first to apply these methodologies to COVID even before the binding information appeared. And his lab is pursuing new directions here. And there's actually a Wiki Corona page out of UPIT that are offering further resources. And I'm sure many more will emerge in the coming days and weeks. And finally, I do want to thank actually the team who is listed over here that provided a very complementary expertise, as well as for their willingness to kind of give up all their life actually for the last 4 weeks to work together on this effort. Our daily meetings were fun and I think resulted in some really quick and very fascinating results but we understand that we have much more to go. And finally, I do want to have acknowledgements to funders as well, to our other work, as well as some of the conflicts we may have. Now, with this, I actually conclude the formal presentation, and we have a number of questions that you have posted. So I'm going to stop the slides and start some of those questions. And I have some of them selected here. One of them, and I'm going to pose this one to Joe from Severick Rende. Is it also possible that a change in the treatment regime could make a difference, such as starting some of the drugs earlier rather than later? Joe.
Yes. So actually, once we have a list that's verified based on in vitro assays, the next plan would be to put the drugs of greatest interest through an in vivo pipeline of human trials. And those trials will probably involve an adaptive trial design, which means that there'll be an overarching IRB approval protocol that will allow us to test the drugs in series with say 10 patients with a particular phenotype with drug A, and then 10 more patients with that same phenotype with drug B. In the course of doing that, we're going to have to carefully define what those phenotypes are. This gets to your question. Sorry for taking so long. And one can think about patients with this disease as falling into 5 or 6 categories. Those who have been exposed who haven't yet converted, those who are infected but are asymptomatic, those who are mildly symptomatic, those who require hospitalization, and those who enter the intensive care unit and/or wind up in the downward spiral with intubation and progressive dysfunction and ultimately death or near death. So each of those circumstances would require different drugs because there are different features of the COVIDome that may be affected by the infection at different stages, which really gets to the importance of trying to gather information on gene expression patterns in these patients at different stages of the infection. We could then correlate that information with the information we have about potential drug target expression or activation or suppression with the use of specific drugs for each of those subphenotypes.
Mm-hmm. Thank you, Joe. Marinka, there is actually a very specific question to you, and I kind of lost it here from Sophia Cricks. And the question is in your AI-based strategy, you mentioned that you generate embeddings by taking the local neighborhood into account. Could you elaborate how exactly those embeddings are generated, and are they reproducible?
Yes, thank you for these questions. So first, embeddings are reproducible. We will be even sharing the drug and disease embeddings, those low dimensional vectors, so that you can also explore them further if you would like them. The way those embeddings are generated are by a method that we have developed that uses graph convolutions as a mechanism, and then optimization to define what is the best way to specify the embedding such that they can really capture topology of the graph. So graph convolutions is the keyword here, and I'm happy also to share some pointers that are also cited in our paper with details.
Marinka, there's another question that you are now on. How does it mean, how does ignoring the direction of interactions when you compute network proximity? I guess that's also for Xiao, but you could also ask in the context of the AI, does directions really matter? Can you actually include them in your approach?
Yeah, so directions from our other studies, we have observed that directions typically matter a lot because they, of course, change the structure of the local neighborhood. In this particular study, for the AI pipeline, we did not consider directions. However, the method could do that if you would have available information on directionality of the edges. What we found out that was really important was to be aware that we have different types of edges, especially because the AI method operated on that heterogeneous knowledge graph. And so it was very, very important that we were not conflating different types of edges. What I mean here is edges that would indicate physical interactions versus edges that would indicate non-physical interactions between proteins, for example. That would really detrimentally affect performance of the AI methods if you would do that. So that was really important.
Thank you, Marinka. There are a couple of bookkeeping questions. Akshay Mohan is asking whether the tools will be available. Many of them already are. I believe Marinka's tools, if you go back to her papers that we had in the slides, are already available on GitHub and other tools. And so are ours. So each of the pipelines actually have already a set of tools that are available. If you do need it, we're happy to set up a site where we put them all there, the links to that. That's something that we can do actually on, on our blog. There was a question, what is the best way of interested medics or managing professionals to contact you? Just send me, for example, an email at barabashi@gmail.com, and I will go ahead and push the email down the team to whoever they actually are in the best position to help you with that. Someone was asking if we could share the slides here. I'm speaking, so I cannot do that, but I will put the link on the social media to the slides for all of you who want to actually follow up on that. So, so don't worry about that. And there was finally a question about, as far as I understand, the interactions from Gordon et al. Are observed in vitro and correspond to which proteins would bind to ones in the cell. Wouldn't it be relevant to first check that the virus can enter the cells in a given tissue first, and so on? And the answer is yes, of course, that would be fabulous. That data, unfortunately, is not yet available. I am sure that actually lots of data like that will emerge in the coming weeks and months, and then we will be able to refine our tools based on that particular knowledge. And this is probably a good moment for us to end also this presentation also with kind of one comment that I would like to add. I think the COVID-19 will turn for us to be a model system for infectious diseases, because this is probably the infection that we have the most data, and we will have the most data ever in the history of humanity, right? So I think many of these questions, probably even when the danger and even when we have cures and so on, will have to be answered. And I think both on the epidemiological area, also in the drug repurposing and the drug development space, this is probably the best map, will turn to be into a best map disease or infectious disease. So we will continue focusing on that, because it offers the wealth of data that if we do well here, we'll be much better prepared for the next round. But in the short run, I think our responsibility and our goal is to work with all of you to take some of these drugs to market and put it in the hands of the patients, those that have efficacy. For that, we need to have both experiments as well as clinical trials, because we want to make sure that this is safe for the patients who are getting it. And that will be actually the question for all of us in the coming weeks and months. At this moment, I want to thank everybody on the team for the fabulous work and their dedication to this, and all of you, and for in attendance, and looking forward to continue the conversation in the coming weeks and months. Thank you.